Hey everyone, it's Patrick Morrison from the Wolfpack Astrobiology podcast, and today I felt the need to address an elephant in the room regarding the metabolism of the first cells. You often hear that ATP is the energy currency of life. ATP stands for adenosine triphosphate, since it's an RNA nucleotide with three phosphate groups lined up on the fifth carbon. The reason it acts as the energy currency of life is because when it reacts with a biomolecule, the phosphodiester bond between the second and third phosphorus atoms are cleaved, and the third phosphate is bound to a biomolecule, converting the ATP to an ADP, or adenosine diphosphate. The phosphorylated molecule is now said to be activated because having an organophosphate group attached will make it more reactive. The molecule will then be fed into a number of metabolic pathways, after which the organophosphate is released as an orthophosphate. This orthophosphate will be inert and can't be reacted with biomolecules to form organophosphates again. To be reused, ATP has to be regenerated by combining the orthophosphate with ADP, and since the phosphodiester bond, which links the two phosphate groups, is a high-energy bond, an input of energy is needed. ATP synthase regenerates most ATP in living cells, and it uses a cross-membrane pH gradient to do so. With a greater concentration of protons on one side of the membrane, a proton motive force, or PMF, would drive any protons on the side of the membrane with a higher concentration to that with a lower concentration should a hole in the membrane open up. The ATP synthase enzyme is embedded in the membrane, and as protons move through a pore in the enzyme complex, the energy from the PMF is used to regenerate ATP. Should enough protons flow across the membrane, there will no longer be a cross-membrane pH gradient and thus no PMF. In order to maintain the cross-membrane gradient, the flow of electrons within other enzyme complexes is used to pump protons from low to high concentrations. The flow of these electrons is driven by the difference in electronegativity between the electron donor and acceptor, in other words, redox chemistry. In us, the electron donor is organic carbon and the acceptor is oxygen. In photosynthetic organisms, these electrons are energized by light. All of this requires complex molecular machinery, which wouldn't have been present when life began. This has led to all kinds of speculation as to how early life replenished ATP, or whether it used a different energy currency altogether. One hypothesis is that the first life replenished ATP, or a similar molecule, via reactions with environmental thioester bonds, or even use thioester groups instead of organophosphates to activate organic molecules. However, this hypothesis hasn't stood up to experimental scrutiny. Another hypothesis states that life emerged in serpentinizing hydrothermal vents. Here, microscale mineral walls would separate the alkaline vent fluid from the acidic ocean water. Crystalline structures were hypothesized to have formed in the mineral barrier that would allow protons to cross over and use the PMF from the environmental pH gradient to produce ATP. However, such a structure would only form rarely, if at all, and not on a reproducible basis, and life has to be able to reproduce. An alternative way modern life can replenish ATP is by substrate-level phosphorylation. Here, biomolecules are broken down into simpler organic ones, and the energy released by this process is used to regenerate ATP. When the organic carbon isn't oxidized, this metabolic pathway is known as fermentation. This is a much simpler substrate enzyme reaction, which could have also been carried out by ribozymes or even magnesium ions. But the revelation that the early Earth had an inert versus reducing atmosphere implies that organic molecules, let alone biomolecules, would not have been common. On top of that, the last common ancestor didn't have the enzymes used in fermentation, making the hypothesis that the first life used it unlikely. In the field of bioenergetics, a major elephant in the room is a mineral known as schreibersite. This is an iron nickel phosphide, and when it's immersed in water, it releases reactive phosphorus species such as pyrophosphate, phosphite, and metaphosphate. This mineral is rare in Earth's crust, but abundant in metallic meteorites. Since the early Earth was pelted with space debris, a large quantity of schreibersite would have been delivered to the surface. The reactive phosphorus species released by its corrosion can be reacted with organic molecules to form organophosphate groups in a manner similar to ATP in living cells. So, early life didn't necessarily have to replenish ATP or any kind of phosphodiester bond. Instead, it could have taken reactive phosphorus species from the environment, used them to activate organic molecules to be fed into metabolic pathways, and, when the organophosphate group was released as an orthophosphate, it could then be excreted into the surrounding solution. One thing to keep in mind is that the late heavy bombardment spanned the time from the first cells to the last common ancestor of everything alive today. The production of organophosphates from reactive phosphorus species is again a simple enzyme substrate interaction, which also could be catalyzed by ribozymes or magnesium ions. Furthermore, since Luca possessed ATP synthase enzymes and an electron transport chain to pump protons, such systems would have evolved while reactive phosphorus species were available as an energy source. In fact, this could be the first mechanism by which reactive phosphorus species were regenerated as the phosphodiester bonds of the ATP molecule. Fermentation could have evolved later as a reversal of biosynthesis pathways powered by schreibersite-derived reactive phosphorus species. Well, that's all I have to say on the topic for now, and if you have an alternative view or something to add, I look forward to reading your comments. Thanks.